So today's lesson is on active transport, and so I want to first begin by recalling what passive transport is. So passive transport involves the movement of materials across the cell membrane where the cell doesn't have to use any of its own energy. Right? And that happens when you have materials moving from an area of really high concentration where they're all crowded together to an area where they're in really, really low concentration. Nature tends to want things to move in that direction, and so that happens passively without the use of the cell's energy. However, having said that, there are times where in our bodies, in living organisms, materials need to be moved where they don't want to go. Right? So, for instance, sometimes you need to move molecules, particles, ions, whatever, into areas where there exist already a number of those molecules, but they still need to go there for a particular function or purpose. So we say that in that case, those molecules, particles, ions, whatever, are moving against the concentration gradient. They're moving from low concentration to high concentration. Unfortunately, nature does not want things to move that way. Nature does not want objects or situations or scenarios to become more organized. And so in order to make that happen, the cell needs to use its own energy. Okay, so that's the main difference between active transport and passive transport. Active transport does need to use the cell's energy in order to move particles around. So a great example of where we actually have this occurring in our body is in the kidneys. Our kidneys, as you may know, filter our blood. And the kidneys are, the way that the kidneys work is that they filter everything out first, and then they reabsorb certain materials that your body still needs. And so often that happens against the concentration gradient because we still need to reabsorb those materials. It seems kind of inefficient, but that is how the kidneys work. Okay, those of you in exercise science may be discussing at some point, maybe you have already, about muscle contraction and how that happens. And so there needs to be the flooding of ions in certain areas in order to make muscles contract. And the last example is with nervous impulses. So nerve impulses need to occur because of sodium and potassium ion channels opening and closing. And in certain cases, we need those ions to move where they don't really want to go. So that involves, of course, active transport, and it uses the cell's energy. So we can broadly separate active transport into two categories: endocytosis and exocytosis. So you may remember from previous science courses where we discussed this prefix endo, and endo means like, kind of taking in. So endothermic, taking in heat. So endocytosis, taking in things into the cell. And that's exactly what it is. It's a process by which the cell actually ingests or takes in materials. So basically, we'll watch a video a little bit later to kind of show this. It's really neat how this happens at the cellular level, and at the molecular level. But the cell membrane essentially folds around a material outside the cell and creates a vesicle. So, and we've seen when we were studying the endomembrane system how that happens, right? So materials might get ingested by the cell, and the cell membrane essentially folds around that material, and that allows it to travel throughout the cell. Sometimes, as an aside, lysosomes may trap materials that are coming in and actually digest them, so whether they're harmful or not useful to the cell, that might happen as well. So when we talk about materials being ingested by the cell, we can further break down endocytosis into two processes. And it's really, in both cases you're taking materials in, but we have different names for processes based on what is being engulfed or taken in by the cell. So the first is pinocytosis. Okay, so when we're talking about pinocytosis, we're discussing the ingestion of dissolved molecules, so essentially like solutions or liquids. Okay, so a great example of that, this occurring in our body, is in the small intestine, where we have cells and that line the small intestine that essentially engulf fat droplets so that we can make use of them and break them down for energy. So we see here in the diagram here, you know, the stars in these squares are representing any kind of dissolved solutes in the extracellular fluid that are going to get engulfed, and then we end up with a vesicle as a result, and that's going to travel within the cell where it needs to go. Now, sometimes we have these larger particles or molecules that need to get engulfed, right? So we saw, I want to say in grade 12 bio, we just saw a video where we watched a white blood cell engulf some harmful bacteria of some description. So that would happen by the process of phagocytosis. So phagocytosis, still ingesting large particles. It's just in this case they're large particles as opposed to these dissolved sort of solutes or ions. And if that's the case, then it's pinocytosis instead. So phagocytosis, just to be clear, involves the engulfing and ingesting of very large particles. And that's essentially what makes it different than pinocytosis. So we've talked about how materials make their way into the cell, but often there are materials that are produced in the cell that become toxic, or they're simply waste materials, and the cell needs to get rid of them. And so that process, the process by which that happens is called exocytosis. So exiting. And I think we've looked at this, you know, we spent an abundant amount of time discussing this when we looked at the endomembrane system. But the vesicle sort of makes its way to the surface of the cell membrane and actually sort of fuses with the cell membrane, becomes one with the cell membrane, and the materials in the vesicle basically make their way out into the extracellular fluid and are transported to where they need to go to be released by the body. Okay, so, you know, as we said, examples of where this is happening or why this is happening is to get rid of waste, certain chemicals we don't need, larger proteins that maybe are, you know, are in the cell and don't need to be there anymore. So a good example of this is the fact that proteins are made in the cell at the ribosome. And the ribosome sends them off to be packaged and modified in the Golgi body. And so the Golgi body, the Golgi apparatus, will eventually pinch off that protein, and it's going to need to make its way in a vesicle out to the extracellular fluid to go and, you know, perform some sort of function within the body. I want to watch a video next that will demonstrate to you what I'm talking about with this business of the vesicles membrane fusing with the cell membrane, and I hope you enjoy.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
So let's watch that again, that little bit about the fusing, and I want you to pay, maybe I'll slow it down so you can see that happening in slow motion. Maybe we'll do that at halftime. Maybe here. There we go.
Contents leave the cell without actually crossing crossing the plasma membrane.
So here you can see how the phospholipid bilayer kind of reorganizes itself to let materials out of the cell. They're going to show us the reverse again. So pay close attention over here and up here. So notice that these, the bilayer sort of reorganizes itself to form the vesicle, and it wraps around. Pretty cool. So the last little bit for you to do is to work on studying questions in section 2.4, those that you were unable to complete yesterday because we hadn't discussed active transport yet. I just want to quickly, before we do that, just kind of compare and contrast active and passive transport. Okay, so passive transport, you'll remember if we just do an overview. So what is this right here? So if you had to guess what this is, hopefully you're all saying in your head, okay, well this is simple diffusion. There's no carrier molecule, a protein, and there's no channel protein. And so the molecules are going straight through. Those are usually really small nonpolar molecules that will travel through. Over here I have my channel protein carrying some ionic or some polar molecules through, and they can do that because usually the inside of this channel protein is lined with some kind of polar or ionic R groups on amino acids. And then over here we have our carrier protein. That was kind of like the elevator on the video that we saw, taking larger molecules like glucose or something like that across the cell membrane. Now remember that these last two are referred to as facilitated transport, facilitated diffusion, because they have these helper molecules helping them out. But none of these three examples require the use of the cell's energy, right? Which is why they're called passive transport. Now over here on the far right, I have active transport. So you'll notice this little thunderbolt. The thunderbolt is there to remind you that in fact we are using some of the cell's energy to take materials where they normally won't want to go from an area of low concentration into an area of high concentration. So against their will, which is why we have to use the cell's energy. If you have any questions, be sure to ask me them on Monday. I'll be happy to answer any points of confusion for you. But I hope this helps.